And welcome to the stage, the last presenter and speaker for this session, Adam Gormley.
Great, thank you so much. I appreciate the invitation for being here. I know this community well, but I don't necessarily know all of you well, and so it's great to have a chance to kind of get to meet all of you. And it's been really fun, actually. So obviously going along with the theme of this conference in terms of autonomous discovery, self-driving labs, I firmly sit in the world of biomaterials and drug delivery. It is a kind of a niche area, but actually an area that I see a tremendous amount of opportunity. You know, for those of you who are not aware, what is kind of biomaterials and drug delivery? Things like the lipid nanoparticles for mRNA vaccination that we all had during the COVID pandemic is very much so an area of active research for our field. Hydrogel, drug delivery, injectable hydrogels, even just like general drug formulations, like protein formulations or small molecule formulations. These are all within the world of biomaterials and drug delivery. In fact, I was actually just in Chicago last week for a drug delivery biomaterials conference. And I can tell you that the world is really ripe for this kind of implementation of these ideas and these methods into this field, because talk about, you know, a discipline that has high dimensionality with this connection to biology. And so a lot of the themes that you've seen here are you know, methods that we're applying. Real quick, I just want to actually give you a quick, actually historical reason as to why I even got here. And this is actually going back to my days of my postdoc. So when I was a postdoc doing kind of work on nanoparticle assembly and developing a diagnostic, I've always done polymer chemistry. I did my PhD doing a lot of polymer drug conjugates. And we were actually developing an assay for polymerizations for diagnostic reasons when we actually saw that our polymerization randomly proceeded in an open vessel, which for those of you who do free radical polymerizations in particular, you know that polymerizations should not proceed in open air. And so without going into the details of why, what we actually discovered was a new ability to do what we now call oxygen tolerant or air tolerant polymer chemistry. And for us, this was really kind of an aha moment. This was like, wow, this is really cool even from a practical perspective, that now we can do this chemistry in just totally open vessels. We no longer have to do this in a Schlenk line. So this is serendipity at its finest. When we did this, so this is Rob Chapman, Cyril Boyer, and I. When we did this, we realized, well, the next obvious thing that you can do with air-tolerant chemistry is now we can do polymer chemistry in everyone's favorite labware, which is well plates, right? You could never do polymerization reactions in well plates prior to this because of the intolerance to oxygen, right? We always do these in Schlenk lines. And what we actually found is that actually, when you reduce the volume of a polymerization reaction as low as the lowest we went, not that we couldn't go lower, was 20 microliters within a 384 well plate, we actually found that polymerizations really like low volumes. In fact, we would get really excellent reaction control, even better. The lower the volume, actually, the better we saw. And so this actually allows you, therefore, to do combinatorial chemistry within well plates. Either 96 or 384 are our most common formats. So this takes me to when I joined Rutgers University. And when I started at Rutgers, my lab originally actually the word automation never existed in anywhere in my faculty applications. But once I noticed that my students were performing the same chemistry over and over again, I realized, you know what? We're just doing liquid handling out of well plates. There are great liquid handlers out there that can pipette in and out of well plates. And so this is when we actually got our first liquid handler, which is right here, to be able to do automatable polymer chemistry. And so we spent years developing our software to be able to enable this. And the basic long story short is that if you want to basically create combinatorial libraries of polymer designs within 384-96 well plates, well, let's just start with the basics. You know, maybe we want to make a homopolymer, a block copolymer, a random heteropolymer. Do we want to control the molecular weight of the polymer? What monomers do we want to incorporate in this polymer? If any of you are familiar with controlled living radical polymerizations, it's actually incredibly versatile. You can actually really mix and match these attributes. We've spent a lot of time also doing automated post-polymerization modifications as well. And all of this is just actually features within an Excel sheet. So every single row is a new polymer, and every single column is all of these features that I described here. And then our software, which we call Polycraft, actually converts all of these designs into liquid handling steps, which our liquid handler implements. Now, one thing that's very important here is our liquid handler is directly controlled by our Python software, and we actually have never used Venus, which is Hamilton's software. So this is just an example. This is actually an outdated video at this point because we actually have a whole new suite of hardware, both the liquid handler, the robotic arms, and things like that. But we still haven't published on it yet, so we're still kind of getting there. But it's a pretty conventional setup. Again, normally our weld plates are polymers, but actually we do a lot more protein formulation, nanoparticle synthesis and characterization, so on and so forth. So this is just a really versatile platform that allows us to do a lot of different types of experiments. And then this is our old robotic arm, which has been since retired. That allows us to be able to transfer these samples to our analytical instruments, such as our UV-Vis. We're about to put our DLS plate reader there as well. And also we have a variety of other pieces of hardware that we kind of swap in and out. You'll see in a second this light box, which I'll talk about in a second, that allows us to be able to do some of this photo-initiated polymer chemistry in a very controlled way. So you'll see that. Okay, so what's going on with that light box? So we are doing photochemistry for the most part. And so if any of you ever do photochemistry, most of you are using a light bulb, right? Or some sort of UV lamp or something like that. And what's the problem with a light bulb? Well, basically what happens is you're not going to get even distribution of light across a weld plate, right? This is actually going to mean that your wells in the center of your weld plate are going to have more light and therefore are going to convert faster than on the outer edges. Right? And also, this is really not tunable, meaning you don't necessarily want every single reaction to receive the same amount of light for the same amount of time. What you need is actually something which gives a little bit more even distribution. And actually, so this is why we created this light box, which we call it a light box. Basically what this is an LED array of 96 LEDs, which means that we can control every single reaction individually. We actually think that this is very valuable, even just to the photochemistry community in general. And this is really how it all started. This was kind of my initial prototype when I first kind of started going down this direction, where we have, again, 96 LEDs and a 96 LED array. Every single LED in this example just turns on automatically. That's just because we haven't soldered on all the LEDs at this point. But it allows us to be able to turn on the light. And what you see in terms of kind of an even distribution of light is you see exactly what you expect, which is that if you look at the lamp on the right, you see a hot spot of light in the center of the wells, whereas as you go out, it gets less and less, it gets less intense, whereas our light box is much more evenly distributed. Now, that was great, but the problem is what we actually want to do is be able to do multiplex lighting, which means that we actually want to be able to control every single reaction individually, as opposed to turning on the whole light box at once or off all at once. So that's when we basically realized there's actually a unique opportunity, especially with our chemistry. I'm not going to get into it. We can actually track conversion of our polymerization reactions by fluorescence. Again, I know you're all chemists, so you're probably wondering why, but for time I don't want to get into it. But when we realize that we can track polymerization as a function of fluorescence, let's see if we can actually, therefore, multiplex this light process. And so actually when we actually upgraded our system so that we, in this example, our newest design, which we're patenting right now, so I'm not going to show it. But in this example, it actually works just like a TV. A TV is just a bunch of flickering LEDs. In this example, it's just a bunch of flickering LEDs that are just being individually addressed and all, again, controlled by our Arduino. And what's really cool about this ability to multiplex this lighting is that, let's say at t equals 0, we've got, I don't know, let's say 60 reactions. So you're seeing here at t equals 0 is just kind of a random distribution of polymerizations throughout the weld plate, because we try to randomize where these reactions are going. And then what we're doing is robotically transferring our weld plate every 10 minutes to our UV-Vis to measure conversion. Basically, how much polymerization has happened. Because some monomers convert within an hour. Some polymers convert in 8 to 12 hours, right? And so again, this is really a big problem, because if you just turn everything on and just leave the light box on for 12 hours, you're going to over-convert many of these polymers. You're going to get a broadening of distribution. So what we really need to do is deliver the right amount of light for the right amount of time. And so what we do is we transfer the light box every 10 minutes to our UV-Vis. It measures conversion, puts it back, and as you reach full conversion, meaning it's kind of flatlining at the top, it turns off that LED. And so what's happening here is over time, 0, half an hour, 1 hour, 1.5 hours, is that the individual LEDs are turning off over time, which means that none of these reactions are being over-converted or under-converted. It's just the right amount of light for the right amount of time. And so, again, I can't necessarily show the latest version because we're still working on getting the patent submitted for this, because we're thinking about commercializing it, because I actually think that the photochemistry community in general is going to find this really interesting. We did actually publish this previous version open source, And one of the problems that we realize with publishing open source hardware, which I actually think is really good, is that probably none of you want to build this on your own. Many people have actually rebuilt our Lightbox, but sometimes actually this thing is really inexpensive. And so if we were to sell it for a couple $100s, you'd probably prefer that as opposed to making it on your own. And so that's why we're thinking commercializing it. But what's really cool about this is that this really allows us to do sequence level control, adding the right amount of monomer at the right amount of time during the course of the reaction, right? This is when you start to get statistical incorporation of individual monomers as the course of the reaction proceeds, because we're monitoring conversion, changing the amount of light. It's a very dynamic process. So that's kind of what we're working on right now. Okay, so what are we working on this with? And so basically, we do a lot of work in protein formulation. And if you ever do protein formulation, one of the things that's really sad about protein formulation is we take these really exquisite macromolecules and then we add really not so elegant excipients to it, right? And that's really a shame because you have this really complex tertiary structure, this really nice surface of a protein, and then you add something like PEG, and I love PEG, but it has a pretty simple structure. And when you add PEG, you are not going to get complementary interactions, right? And so what we really believe is that what you should be doing is fitting the excipient to the protein itself. This is the equivalent of making sure that you have a tailor-fitted piece of clothing for your body that fits your body, as opposed to a one-size-fits-all excipient that just works for all proteins and isn't always the case. This is a really high-dimensional problem. How do you design a polymer excipient around the protein? And we've been partnering with Mike Webb at Princeton University, who I know many of you all know, for the last couple of years. Now this work is a few years old, but I wanted to incorporate into this talk because I don't really know all of you that well. But what we did with Mike Webb is we basically said, okay, let's do active learning. Now that we have this ability to do automated polymer chemistry, let's do cycles of design, build, test, learn with active learning, and maybe just look at 3 kind of general enzymes here, just horseradish peroxidase, glucose oxidase, and lipase, and then iterate throughout the design space, changing the properties of the polymers, and see if we can find what polymer fits these specific proteins. And so I'm really glad, actually, we have a new liquid handler now. Our old one is quite old. Protein formulation, we do tons of protein formulation beyond this project, requires a lot of reagent transfers all the time. And so our new liquid handler is actually much faster. This is still, I think, 1.5x or 2x speed, so it's not actually that fast. But it's much faster at being able to allow us to do really rapid combinatorial protein formulation experiments. And I don't need to belabor this point because you all know this very well. All that the point of this slide is just to suggest that we are following really tried and true garden variety active learning Bayesian optimization experiments through parameter space. And so this is what you actually see. So this is multiple iterations of design, build, test, learn. We initialize the model with a seed library. And then what you're seeing here in the black for each generation is actually the centroid of the data. And what you're seeing is that the machine learning model is navigating through this parameter space, balancing explorer and exploit, as you all know. And what's really cool is actually watching this go along. And sometimes you actually look at the designs and you say that doesn't make any sense, but I'm not going to bias you, so proceed. And it's actually really fun to watch it jump through different parameter spaces. We do this a lot now, and what we actually find is that we actually don't need that many generations. These things are learning very, very quickly. And so we actually generally don't even do 5 generations anymore. And what's really great, and I love this is my favorite figure of all time, is you actually get fully mapped structure-function landscapes, right? You always think about structure-function landscapes as a free energy diagram, right? And this is about as high resolution as you're going to get from a protein formulation problem. This would be incredibly hard to do by one experiment at a time as a human. And what you see is that in horseradish peroxidase, the structure-function map is incredibly diverse. There's a lot of local minima. And so that would be really hard to figure out. Whereas in lipase on the right is actually kind of converging on a single solution, meaning it is this balance of properties that are really going to give you the results that you are looking for. And so again, horseradish peroxidase is diverse. Lipase, there's actually really one big solution that worked really well, kind of a really deep minima. But that's not something you would have been able to figure out one experiment at a time. And this is just to show that, basically when we do this process, we are getting very, very specific excipients, meaning that if you take one excipient that works for horseradish peroxidase and apply it to the other enzymes, does not work. These are not one size fits all, very fit for purpose. And I really truly believe that, obviously there's regulatory concerns about, developing novel excipients for new drugs. But, in speaking with people who are in pharma, if you can enable a new drug in a way that wouldn't be possible without this novel excipient, then maybe it's worth it. And I'll actually give you an example of this. Spinal cord injuries, or even traumatic brain injuries. The biggest reason why you cannot recover after a spinal cord or a traumatic brain injury is because you literally get a physical scar. This is physical scar tissue. Your astrocytes basically secrete a bunch of chondroitin sulfate proteoglycans, and these CSPGs basically just physically limit neuronal outgrowth because it's just a physical chemical scar. Okay, so what do you do about that scar? Well, there's actually a really great enzyme out there that the TBI and CSI community have been known about for a really long time, which is chondroitinase ABC. It's this enzyme that is derived from bacteria, and it is really good at breaking down chondroitin sulfate proteoglycans. Really good at it. It's just amazing that this bacteria figured out this enzyme. So why haven't we solved spinal cord injuries? Well, the reason is because this is the activity of this enzyme over time. 100% at time equals 0, put this at 37 degrees C body temperature, which the bacteria did not evolve in, and activity drops down. Actually, within an hour, it's actually totally dead. So if you inject this drug into a spinal cord injury, within an hour, this drug is gone. Great activity, very poor druggability. So when we did all of this, we said, this is a great use case scenario. Let's figure this out. This is one of those examples where this drug has been worked on for decades, but has never been enabled. And so we said, let's see if we can figure out an excipient that works for this enzyme. So we did everything I mentioned before, and we saw two really important things with this, a couple polymers, but this one polymer in particular. Number one, we tripled the original enzyme activity. So 100% is native enzyme activity. Time equals 0, we have 300% enzyme activity. We see this time and time and time again. We are amplifying, and when you find the right excipient for the right enzyme, we are amplifying its activity. We have a couple hypotheses as to why. We're pretty sure we know why, but we see this resoundingly, almost always. It's really cool. The other thing is that although obviously the activity does decay, pretty quickly after a few days, but after a few days, this thing's still alive. In fact, even out to 1 week, this enzyme is still alive. You compare from an area under the curve of a druggability from 1 hour to 7 days, that is huge. And so we were really excited about this. We have run into all sorts of new issues as to why this doesn't work, mainly because if you inject anything into the spinal cord, actually our spinal cord and our CSF are under positive pressure. It actually clears drugs out really quickly. So now we actually have to encapsulate this into a shear thinning injectable hydrogel and get this thing retained. But we do see that, we incorporate into a hydrogel, we see retained activity, and we can actually see release. Right now our best is about 3 days. We're actually getting sustained release of this active protein, which is really great. So we're pretty excited about this, and it's a big area of active research. We think that this is exciting, not just from an academic perspective, but also from an industrial perspective. So we have this startup company called Pleximer. Just because of this community, I just want to point out the logo. Every compound is unique, every fingerprint is unique, and that's what the logo is all about. It's actually a fingerprint out of polymers. So this is the ethos of our startup company. And we're actually not at all supported by investors. We're entirely supported by corporate contracts. Because industry is actually really liking this. And so we're pretty happy to be growing this year as a result of non-dilutive growth just from actual industrial interest. Okay, let me take a second. The world of biomaterials and drug delivery, I am feeling very alone, to be honest with you. I was just mentioned, I was at a biomaterials conference last week. Nobody is doing this stuff. Now, first of all, I'm very happy to be kind of in this niche space, obviously. But at the same time, I'm kind of like, come on, people. Like, these are really powerful tools. Like, I know I'm incredibly biased, but like machine learning, I mean, at the very least, we should be doing this as a community. And it is really hard to even fill a single half-day session at this conference last week on machine learning. In fact, we haven't been able to do it for years. And so why? Why are people so slow to uptake on this? And so it's because this is actually quite difficult, right? There's a lot of things involved. And so we realized there's a couple barriers to entry to bring people along with us. And so one of the first things we said is, frankly, they don't know machine learning, which is understandable. I couldn't even define what machine learning was 5 years ago, right? And so there's a lot of users guides out there for machine learning. I'm certainly not unique in that regard. So we created this users guide in response to limited uptake by the community. And this was obviously specific for polymeric biomaterials. one thing that stands this user's guide out maybe a little bit more relative to some of the other ones is the fact that we actually published all of our code in a Google Colab notebook. So maybe some of you use Google Colab. One of the things about these kind of online Jupyter notebooks is that they're just really easy to use. I mean, you don't have to worry about all the little nuances of Python that kind of frustrate you, like, versions and whether packages are installed necessarily on your local computer. And so we actually just put all of this information just on a Google Colab notebook. And so the user's guide itself walks you through the theoretical understandings of what machine learning is, but then the Colab notebook in a very, very commented way starts by, okay, let's first start by importing your data. Okay, now let's inspect the data, see if there's any missing data values. Is there anything that's wrong there? Okay, now let's scale the data. Now let's, train, test, split the data. Now let's fit the model. Now let's do kind of model validation. And then maybe ultimately we're going to do, SHAP analysis on all this stuff. And so, we really basically said, in this user's guide, please feel free to start just actually making a copy of this Colab notebook and import your own data, and then start playing with these tools. Last I checked a couple months ago, actually, we've had over 10,000 accesses in the first year of this being published, which meant that we really tapped into an unmet need within our community, which I was really excited about. Okay, so then what's the problem? Okay, that's great. Now people are hopefully getting into machine learning. Then the automation side of things. Immediately, it's a full stop, right? Because automation is actually quite difficult. And the other thing that people say is that automation is very expensive. So here's Polybot up there, a little plug, right? And I don't necessarily mean to say that it is the most complicated system, but it is on the complicated, expensive end of the spectrum. And not every single lab, especially a graduate student, if you're the PI of a lab, maybe you're not going to go full automation, but maybe there's that one PhD student in your lab who says, I'm interested in it, but my lab doesn't have the resources or expertise to try it. So how do we go about actually making, even from an automation perspective, this accessible to the average person? And OpenTrons has actually been really good about that, right? OpenTrons was built on this vision of making sure that some things like liquid handlers, like you can buy an OpenTrons liquid handler for $7,000. I mean, the costs have gone up over time, and that's accessible to a lot of labs. But it's not accessible to every single lab. And so we thought that there's actually maybe an area of even less expensive kind of open hardware, open source, that can make this even more accessible to the average student with a very limited budget. And that's where we've created what I'm calling the minimum example of a self-driving liquid handling lab. So what do you need in order to be a liquid handling lab? Well, you need an XYZ, stepper motor system, and you need something that can pipette, right? Like a syringe pump. And so that's all. And so we thought, what is the least expensive version of an XYZ stage? And it turned out to be just an XYZ plotter, like a pen plotter. You guys ever use these things and can draw? It can you attach a pen to it. There's this great one called AxiDraw. It has actually a Python API. You can literally just give it an XYZ coordinate and it will go there. It can lift it up. And then all we did was basically just connected that to a syringe pump. Many labs have syringe pumps laying around. That's not an unusual piece of equipment. And put it all together and created this liquid handling lab. The syringe pump is like maybe $1,000. The AxiDraw is maybe $500, but that's it. That's not that bad. And so we've actually been using active learning. In this example, we've just been optimization of red, green, blue. See if we can do active learning so if we can optimize for specific RGB colors. This is just food coloring. And actually just putting this on a UV-Vis plate reader. Again, I'm sorry to say a UV-Vis plate reader is maybe a bit expensive, but most labs have UV-Vis plate readers, so not a big deal. And RGB, obviously very predictable, figured things out pretty quickly. What we've actually been doing is been using this to optimize enzyme assays, which is actually something that is useful. And so we're writing right now part 2 of our user's guide. It's going to be called, at least the current draft of it, as we're writing it, is going to be called A User's Guide to Your First Self-Driving Liquid Handling Lab. It is going to be fully open source software. It's going to show you how you do active learning. Mike Webb right now is writing the active learning tutorial piece of it to actually show people how to do active learning. And so this is hopefully going to give people from a hardware and a software perspective the starting point to doing autonomous experimentation in liquid handling environments. I am an engineer doing chemistry. That's how I always have been. And so I love automation. I just have been really getting into it. A couple things I've been doing just on my own, just to keep myself happy, and rather than writing grants all the time, is been building some of our own hardware. We do a lot of our own hardware and electrical design. I actually built this liquid handler, semi-inspired by the Jubilee platform on the left. I actually built this at home in front of the TV in my den. Because of that, my 4-year-old, some 4-year-olds love dinosaurs, some 4-year-olds love airplanes. My 4-year-old loves robots because I've been building this robot at home and has been really loving it. So everything's about robots. But it's really cool. Actually, I gave this to my startup company because we didn't have the budget to have to buy our own liquid handler, and it's been operating ever since. So this little thing that I built at home has been pretty straightforward. But the other thing that I'm getting really into, and I was talking about this right after the break, is I'm really getting excited by the value of these robotic arms, these collaboratory robotic arms. Couple that with machine vision. Holy cow! I am seeing tremendous potential. Everything that liquid handler on the left is doing can be done by that. Actually, almost every single thing that we roboticize can probably be done by a robotic arm, as long as you incorporate machine vision to all of this. Be able to put labware in front of it, be able to do object detection and coordinate evaluation, and then be able to do end effector swapping and be able to go from a single channel to a multi-channel to a gripper to a mixer. I think robotic arms can do it all. Actually, I think it can do almost everything. And if you have multiple robotic arms, it can do a lot. So this is an area that I'm getting very excited about. And this is why, actually, this is just sitting on my desk at work right now in my office. This is just doing kind of a classic pick and place type of algorithm. I'm really enthusiastic. I'm loving this right now. I'm having a lot of fun and probably not spending enough time grant writing. But anyways, that's it. I appreciate the opportunity to speak in front of all you all. I'm hoping the world of biomaterials and drug delivery picks up this a little bit faster. But honestly, you guys are really the ones, the organic chemists in particular, are the ones who are really driving this. So I'm inspired by so much of the work you're doing. I'm trying to learn from you all. Again, I'm like an engineer, a biomedical engineer, trying to learn from you guys who are really kind of pushing the boundaries for this. So I'm always trying to play catch up with you, but then also trying to bring my community with me as well at the same time. So anyway, with that, I'll thank you all. Thanks, Mike Webb, my major collaborator through all of this, my students, my funding for supporting this work. Thank you very much.
The question is just to acknowledge and appreciate your efforts to ensure that you democratize all this complexity. So thank you for that.
Thank you. Appreciate it. We spent a lot of time working on it, but I think part of it started off just by having fun and doing this for the fun of it, and then realizing, oh, actually, this is maybe also useful to others.
Yeah, so to follow up on the ARM vision, I think that's really neat. So everyone's trying to make humanoid robots these days, but you maybe just need the arms. You don't even need actually anything humanoid. But the part of it that wasn't clear to me is, to what extent are you also moving towards sort of fuzzier natural language descriptions of the experiments that you're trying to do versus hard-coded expert workflows?
So that's exactly why that robotic car is on my desk. I'm realizing the value of these types of tools. So our software to control overall automation includes a 65-page user's manual that I wrote. And that's context. That's a fabulous context for something like an LLM to be able to access. And so I actually really think, when I think about what Opentrons I should be doing, if Opentrons is not doing this, I think I'm pretty sure they're doing this. I feel like you should be able to go up to an Opentrons, place labware anywhere in front of it. Like, you don't have to teach it positions. It should just have a camera to say, oh, that's a well plate. Now I see there's a well plate there. Now I can calculate its positions. And then you should be able to say, make a serial dilution that does this. Why is that? Not only is that possible, that's extremely achievable. Right? Now you start to go further beyond that and start to say, give it a whole bunch of context, like a 65-page user manual, and maybe a whole bunch of examples of a bunch of different experiments. So the first example I'm going to do when I get back, we're going to do a hydrogel release optimization experiment. We can just take a 4-arm peg, that cross-links, encapsulate a protein in it. I should be able to say, hey robot, you've got a peg in vial 1, the crosslinker in vial 2, and protein in vial 3, and you've got your plate reader next to you. I bought a little cheap one. Make a combinatorial library of hydrogels and then take time points every single hour for the next 8 hours, put it on UV-Vis, and tell me the release profile of that combinatorial library. That's extremely doable. So that's what I'm doing right when I get back.
Yeah. Yeah, I'm not as optimistic as you that OpenTrons is doing it. Everything else, I agree with what you said. I think you should build it.
So, yeah, I thought about that. Good question. I met the CEO of OpenTrons.
There might be an opportunity there, yeah.
If they're not doing it, I mean, there's no reason why in 2025 there should be taught positions. Teaching positions, the grad students in the room, I'm looking at you. How annoying is it to constantly update positions and things crash? There's no reason for that. The camera should give you real-time object detection and tell you that is right there. I'm telling you, it works easily.
I had a question. When you showed these schematics of protein, when you showed these schematics of the proteins and the excipients binding, so with the functional assays, were they all just based on protein function? Did you have any molecular dynamic simulations or other molecular modeling to kind of show the picture of this really tight fit of the protein that the polymer of the protein that you're kind of proposing?
So that is actually part of this big, larger collaborative project. It's not just Mike Webb and I. It's actually a few different people. One of them is someone who specializes in MD simulations. So they've been doing that, basically trying to rapidly simulate the polymer-protein interaction and actually see if we can use that as feedback to the model in case those in silico predictions, simulations, can help inform the experimental workflow. So that is something we're working on.
Good. All right.